Hello friends, let us now take up the processing and preservation of fruits and vegetables. In this week, we will devote 5 lectures on fruits and vegetable processing. So, in this first lecture of fruits and vegetables, let us study that principles of shelf life extension of fruits and vegetable, particularly the respiration and ripening processes. Just in this slide, I have tried to give you an overall picture or current scenario of fruits and vegetables in India. To look here in this first picture, that production in million metric ton is given from 2009 and onwards. Total production in the year 2016-17 of vegetables in India was 176,177 million metric tons, and this accounts for 4.2% rise over the production that was in 2015-16. Similarly, fruits total production was 93,707 million metric tons in the year 2016-17, and this was the about 3.9% rise over the production that was done in 2015-16. So, the production scenario shows that on the basis of that, and the availability of the fruits and vegetable is calculated. So, obviously, on the basis of the production, projected availability is much high than that which is recommended for consumption. So, as for our production statistics, we are self sufficient, we have sufficient amount of fruits and vegetable even which is required for our daily consumption. But actually, if you see the data says that the actual consumption is less than that of the recommended consumption. So, the main reason for this is the losses which occur in fruits and vegetables. That is, data says that on an average around 18 to 20% of the produced horticultural produce, fruits and vegetable gets destroyed in the value chain. And it results in the losses of the material, it results into the significant loss in the economic terms in the country. The data says that more than 50,000 crore rupees worth fruits and vegetables are annually lost from the country. Export is significantly less. Of course, in the recent years there have been increases in the exports of fruits and vegetable and their products, but at present the major focus is to bring down the losses of fruits and vegetables. And when we bring down these losses, it may result into availability of the more material. So, actual consumption may increase, even the export of the processed fruits and vegetables or even the fruits and vegetable whole fresh fruits and vegetable may also increase. So, the major processes controlling the fruits and vegetable, their shelf life include the respiration and ripening. So, let us now understand what are these processes and what are the various factors which govern these processes. As far as the respiration is concerned, it is a metabolic process that provides the energy for plant biochemical processes. Various substrates used in important metabolic pathways in the plant are formed during respiration. Aerobic respiration consists of oxidative breakdown of organic reserve to simpler molecules including carbon dioxide and water, and there is subsequent release of energy. The organic substrates broken down in the process of respiration may include carbohydrates, lipids and organic acids. The process consumes oxygen in a series of enzymatic reactions. Glycolysis, the tricarboxylic acid cycle and the electron transport systems are the metabolic pathways of aerobic respiration. The respiration rate, at what rate the respiration process is taking place, that will influence both the ripening process as well as the shelf life of the fruit or the changes which take place in the fruits and vegetable. So, study of respiration rate becomes very important aspect. So, the rate of respiration is expressed either in terms of O2 consumption, that is the consumption of oxygen, rate at which the plant materials consume oxygen, and or the rate at which they produce carbon dioxide, because these plant materials they take oxygen and give out carbon dioxide. So, the usual methods of respiration rate determination include the closed system or static system, the flowing or flushed system and the permeable system. In the closed system, as you can see in this figure, that there is a container, gas tight container of known volume. It is filled with the required commodity, fruit or vegetable or other plant commodity of which we want to measure the respiration rate, and inside the container obviously the ambient air is the initial atmosphere. After putting the product in the container is closed, it is made airtight and it is connected to the head space gas analyzer. So, the changes in the concentration of oxygen and carbon dioxide over a certain period of time are measured and used to calculate the respiration rate as per the equation that is provided here in the slide. In the other method, that is the flow through system method, the product is enclosed in an impermeable container through which a gas mixture flows at a constant rate. The respiration rates are calculated from the absolute difference in gas concentrations between the outlet and the inlet when the system reaches a steady state. And there are the expressions for respiration rate of O2 or respiration of rate of CO2 are given, that is they are calculated from the absolute differences in the gas concentration. In the permeable system, a package of known dimensions and film permeability is filled with the product, and the steady state concentration of oxygen and carbon dioxides are determined, and a mass balance is performed on the system in order to estimate the respiration rate. So, here basically the mass balance of at certain time intervals is the major criteria for the determination of the. So, after having studied the respiration rate either on the basis of oxygen consumption or on the basis of carbon dioxide evolution, another important parameter is determination of the respiration quotient, and this is actually, as you can see here in the figure, RQ is equal to RCO2 by RO2. So, this is the ratio of the carbon dioxide produced to oxygen consumed, and the respiratory quotient is normally assumed to be equal. That is, if the metabolic process or metabolic substrates are carbohydrates, RQ should be 1, because the total oxidation of 1 mole of hexose consumes 6 moles of oxygen and produces 6 moles of carbon dioxide. So, there is a equal O2 and CO2 ratio. If the substrate is a lipid, the RQ is always lower than the unity, because the ratio between C and O in lipids is lower than the ratio of C and O in carbohydrates. If the substrate is an acid, the RQ is higher than the unity. The normal RQ values in the literature are reported as ranging from 0.7 to 1.1 for horticultural produces. The RQ is much greater than 1 when anaerobic respiration takes place. In the anaerobic respiration or in the fermentative metabolism, ethanol production involves decarboxylation of pyruvate to carbon dioxide without oxygen uptake, and therefore, the respiratory quotient is much high. Horticultural produces can be divided into 2 major groups according to their regulatory mechanism underlying their ripening process, which majorly govern their shelf life. You can see here in this picture also, that is on the basis of their respiratory behavior and the rate at which they respire. The fruits and vegetable and other horticultural produces are grouped into climateric and non-climateric. The climateric commodities continue to ripen after harvest. Examples are banana, mango, papaya, avocado, guava, etc., etc. Non-climateric horticultural produces generally do not ripen after harvest. An example of this category are citrus fruit, strawberry, grapes and almost all vegetables. Vegetables, this vegetable term include a great variety of plant organs like roots, tubers, seeds, bulbs, fruit, sprout, stem, leaves, etc. And therefore, they have different metabolic activities and consequently, they will have different respiration rates. Even different varieties of the same produce can exhibit different respiration rate. So, various factors that affect the respiration rate of horticultural commodities include temperature, type, variety and maturity stage of the commodity, their climatic behavior, presence or absence of oxygen, carbon dioxide and ethylene or other growth regulators, and other growth factors such as stress, injury, etc. to the fruit or vegetable or to the product. In general, you can see here in this picture that the non-climatic commodities have a higher respiration rate initially. These dotted lines are showing that the respiration rate as well as ethylene production of non-climatic commodities. So, initially non-climatic material has high respiration rate, and this with the time after harvest the respiration rate decreases, and the ethylene production, which is a ripening hormone, is almost constant. Whatever ripen hormone ethylene was getting produced at the time of harvest, it almost the same concentration is maintained. But that in the case of climateric materials, their respiration is initially high, but it decreases, comes to a climateric minimum that is the climatic minimum here at the target, at the point when you see ethylene production also. If you see in this case, it was initially less and then it increases, means that there is a coincidence sometime that is the climatic minimum it reaches, at that time the ethylene production increases, and that is the point where the ripening of fruits, ripening process of the fruit starts, and this respiration, and because of this there is a significant increase in the respiratory behavior of the climatic fruits. It reaches to a point that is the climatic maxima. This point is known as climatic minima, this point is known as climatic maxima, and during this period of climatic rise, there are several significant changes which are brought in the fruits and vegetables, that we say that the fruit is ripening, that is the ripening process of the. We will come to this aspect little later. So, and at this point when you can say that at this point the fruit is of maximum eating value, and here the senescence sets into. At this climatic minimum point, the ripening starts. At the climatic maxima point, the ripening stops and the fruit has maximum eating value, and at this point it starts spoiling, that is senescence sets into, and finally after some time. So, there is a relationship between the ethylene production and ripening and finally the senescence. So, climatic producers exhibit a peak of respiration and ethylene production associated with the senescence and ripening. I already explained to you. So, now that is the respiration, as you have seen in the climatic fruits, respiration is that is associated, it results into the ripening that period. So, ripening is the phase in maturation process of fruits and vegetable. As the fruits mature, their tissues undergo innumerable changes ranging in the change in the texture, color, aroma, composition and other components of the material, including the nutritional changes. The term ripening and maturity are sometimes used alternatively and are regarded as the state when the fresh produce becomes appropriately suitable for harvest as well as for eating. Though the degree of ripening or degree of ripeness has remarkable clarity in terms of visible changes including color and other attributes, it is sometimes ambiguous to define the right stage of desired ripeness of the fresh produced depending upon the human choice. As in the case of fruits like tomato, ripeness may depend on the degree of sweetness or development of acidity an individual may find attractive. Some people might like sweet taste of the tomatoes, some people like good red color of the tomato, some people like that acid of the tomato. So, and these colors and acid etc., are formed during ripening process. So, it depending upon the individual perceptions, one can accordingly define that what is the final or correct stage of ripening. In contrast to the fruits, the vegetables do not possess any obvious change and maturity is exceedingly difficult to define. However, vegetables during the maturation period exhibit changes in the chemical composition, physical structure, textural changes, some softening etc., may take place sometime. Lignification or hardening of the vegetable also may take place. So, the changes that take place during ripening of the fruit are those in the color, there may be softening and associated alteration in texture, production of volatiles and flavor components, changes in sugar and organic acid metabolism. So, that is the sour fruit may change into a sweet, intensely sweet commodity or fruit after ripening. Changes in the membrane permeability may take place, which releases compartmentalized enzymes, and these enzymes that is facilitate the ripening process or other physiological processes. Even there may be some increase in the enzyme synthesis, new enzymes or new hormones etc., also might be produced during the ripening process. Other changes associated with the ripening may include changes in carbohydrate, pectin, organic acids and so on. So, the respiration rate, as I told you earlier, is directly related to the shelf life of the materials. In this graph, I have shown that that is shelf life of some fruits or vegetable and their relationship with the respiratory. You can see that is in the x axis it is the respiration rate, from low to high it is going. So, the mushroom which has got a high rate of respiration, it has comparatively lower shelf life. In the y axis is the shelf life from short to long. Potato, you can see the vegetable and the extreme ends of both, that is in the y axis, that is the potato which has a comparatively lower rate of respiration among all these commodities which we have taken here. So, it has a low rate, so it has high or long shelf life. So, now after having this brief understanding about the respiration and ripening processes, let us see that what are actually the problems in post harvest storage of fruits, or the vegetables. So, in this slide we have seen that is a respiratory behavior, and you can see that as I explained earlier, that this is the climatic minimum and this is the climatic maximum. At this point the fruit has got that is the ripening starts. Here it is completely ripened, all the changes associated with the ripening are completed and the fruit is in good state of quality state, right. So, you can say in this case that this line, this is the shelf life of the fruit, that is here this is the period after definite period after harvest and before the consumption, during which the food is shelf life. But what happens that during this when the fruits and vegetables ripen, that is depending upon the, as you have seen in the earlier slide, this first the commodities which have high respiration rate, they have short shelf life, and the short shelf life leads to the spoilage of those material. So, somehow by appropriate means we need to increase the shelf life, means somehow by one or other means we have to decrease the rate of respiration, right. So, for doing this, even that to decrease the rate of respiration and to make sure that the fruits and vegetables they have little more shelf life, this industries or farmers or other agencies, they treat with some growth regulators etc., that is the hormone which are ripening hormone, ethylene which produces here, then they treat the commodities with this, and in the process sometime even synthetic fungicides like benomyl, thiobenzidazole etc., are treated. So, these are carcinogenic material and they produce some undesirable changes, so shelf life may increase, but this the product commodity may not be safe for the consumption. So, the normal effort is to manipulate the respiration rate of respiration by other means, like we have seen that is the factors which influence the respiration rate are the gaseous composition temperature. So, particularly the respiration rate of the fruit decreases with the decrease in storage temperature, that it decreases with decrease in oxygen concentration and increase in carbon dioxide concentration of the storage environment. So, by manipulating the carbon dioxide and oxygen compositions in the environment surrounding these fruits and vegetables, as well as by manipulating the temperature, lowering down the temperature, we can reduce the self life, you can reduce the respiration rate, and accordingly that is this life which was here, that is the slope of this life can be reduced, and therefore, the life can be extended from this point you can see here this. And that is the basic principle which is followed in the for the extension of self life of fruits and vegetables. Appropriate methodologies are used to manipulate the respiration of fruits and vegetable after they are. So, I will come little later to this. Before that is if the fruits and vegetables they are not stored in proper manner, it may result into different disorders, metabolic disorders, and one major disorder is the chilling injury, that is the exposure of the climatic fruits to low temperature storage below 13°C or that causes chilling injury. Even the banana etc., banana if it is put into the refrigerator, blackening of the banana may take place. So, similarly there are many other examples. So, the primary reason caused for chilling injury, that is the primary injury may be because of the two processes mechanisms, that is excess production of ROS including superoxide anion, hydroxyl radical, hydrogen peroxides etc., which further increases the production of superoxide anion. The accumulated ROS changes the membrane organization, such as decreases in the unsaturated fatty acids of the membrane lipids and accumulation of membrane lipid oxidation related malonaldehyde byproducts etc., and these result into the visible changes on the surface of the fruits etc. Even enzymatic oxidation of phenolic substrate by PPO to form black or brown products are the other mechanism which cause chilling injury. And in this you can see the different storage disorders of fruits like soft scale, low oxygen injury, internal CO2 injury, superficial scaled, low temperature breakdown, external CO2 injury etc., how they look like, right. So, in fact, for extension of self life, as I told you earlier, that is there are different methods. In fact, here the novel method I will be discussing for that, and in all these processes in one or the other way we manipulate the rate of the respiration. So, the methods for manipulation of the rate of the respiration of fruits and vegetable, and therefore, for extension of self life includes modified atmosphere packaging or control atmosphere storage, intelligent packaging, active packaging, edible coating etc. So, see these are the some of the novel methods. So, although in the next lectures in this series we will be taking up these topics separately, some of these topics, but today I will just like to give you briefly the concept in each case. Modified atmosphere packaging or control atmosphere storage, as I told you, it is the changes in the gas composition. Normally oxygen is lower down and carbon dioxide is increased. Control atmosphere is the alteration of the natural gaseous environment and maintenance of this atmosphere at pre-specified conditions throughout the storage time. whereas the modified atmosphere is the initial alteration of the gaseous environment in the immediate vicinity of stored and packaged product. Modified atmosphere packaging, as you can see here in this picture, we saw, I explained you that when the commodities they respire, they consume oxygen, they give carbon dioxide and also some water is produced, so they give out water. So, it means that in the atmosphere in the vicinity of the fruits and vegetables, the environment changes, oxygen normally becomes less, carbon dioxide concentration becomes more and water concentration becomes more. So, what is done in modified atmosphere packaging? Having appropriate means, suitable packaging material or other film like semi permeable films etc., are taken to pack these fruits or vegetable, all right, and these semi permeable membrane, that is they have that is we need to select it in a proper manner, which having suitable permeability that is there to oxygen, permeability to carbon dioxide, their permeability to water vapor. So, that they allow that is the control transport of these gases in and out of the, and finally, we by this process we maintain a desired O2, CO2 and water composition inside the environment around the packet. So, that is the concept of modified atmosphere packaging. We will take up this further in the next lecture in details. And a combination technology, another novel concept, that is the combination of active packaging and control atmosphere storage. Let us see that if in the normal atmosphere there is about 20.9% oxygen and 0.03% carbon dioxide, and we want that for the control atmosphere there is about we want that oxygen should be maintained to 10% and carbon dioxide should be maintained to 10%, means that is carbon dioxide has to be increased from 0.03 to 10 and O2 has to be brought from 20.9 to 10. So, it means that the concentration gradient is very high. It may take a long time for the establishment of desired environment inside the package, and therefore, it may take even longer time for equilibration, and during this period itself the fruits and vegetable may have significant changes, its quality might have changed. Another case, if you see that there is a condition where the by other means the atmosphere surrounding, that is the atmosphere gaseous composition in the storage atmosphere is changed to 5% O2 and 10% CO2 by some mechanical means and all those, right. And so, in this case that is 10% CO2 is already maintained. So, it is there the 10% O2. So, from 5% to 10% we need to bring. So, here it becomes easier, that is the since concentration gradient is less, it is low in O2 may be possible and high CO2 may be maintained. So, it takes a short time comparatively, in lesser time the equilibrium required equilibrium gaseous composition etc., is achieved. So, the material quality, and so it has both together, it has a we get the comparatively higher shelf life, longer shelf life of the material. Similarly, there is a edible coating technology, we will also take up in the active packaging. We are using impermeable or selectively permeable materials, packaging material we are packing. Here in the edible coating, some food grade materials etc., are applied on the surface of the fruit or vegetable. The basic principle here is also to make sure that the movement of the gases, that is oxygen from outside to inside the tissue and CO2 from inside to outside the tissue, that is controlled, and accordingly the rate of respiration is controlled and shelf life of the edible coating also we will take up separately the details. Another important concept is equilibrium humidity packaging. As you have seen in the respiration rate, that this when they respire, they release CO2 and H2O, water. So, this water inside the packet and water outside the packet, that is humidity inside the packet, the environment inside the packet as well as in the environment outside the packet, in the storage environment. One should have proper equilibrium, that is it should equilibrium. Otherwise, if the equilibrium outside the storage environment is more or inside the, because inside this when the fruit is respiring, water is getting released, and if the outside is also more, then water from inside it may not go out, or if it is inside it is less. So, from outside it may come in. So, it may result into the condensation of the moisture inside the packet, and this condensed moisture may act as a vector for the bacterial growth and fungal growth, etc., and it may result into the spoilage of the fruits and vegetables. So, it is a very, very important aspect that the packaging material, etc., are taken in such a way, calculations are and it is actively packaged, moisture scavengers, etc., taken to consider, and efforts be made that the relative humidity of the environment outside the package, inside the package, around the fruit, there is a equilibrium is maintained, and if the equilibrium is maintained, it will avoid the condensation of the moisture. Okay. So, to design equilibrium humidity packaging for fruits and vegetables, it is essential to have the information about the rate of water evaporation from the fruit surface, or you can say banana surface. We have done studies on this aspect in banana and guava, etc., to large extent. And so, it should be balanced with the water vapor transmission rate of the packaging film, making the equilibrium humidity packaging, and it gives better shelf life of the. So, with this I thank you for your hearing. In the by now, I hope you have got a good understanding about the ripening process, respiration process, how fruits and vegetables get spoiled, etc., and what are the different methods by which their shelf life can be increased. So, in the next classes, we will take up these methods one by one in little detail. Thank you.